glad to be here celebrating this meeting. This meet the scientists specific about the environment and biodiversity issue. Let me introduce first Gabriella Kakonyi. She is the managing chair of the outreach activities of the very nice, very wonderful, interesting project called Outreach. And she will present the rest of this meeting.
So, hi everybody. So we were working in a sustainable municipal remediation project. Here we are glad that Gabriella, she's doctor and she's our project manager. She will introduce us about our project and the topic about our project. And then Natalia, Fernandez, and Vera, they will be speaking about some areas like transportation, transport, and Bacterial. I don't know. So let's go with it.
Thank you. So am I standing okay with the camera? Is it going to be right? The other thing I wanted to ask is, can you hear me? Because I'm not
you're gonna ask to shoot that off if possible.
Tienen un micrófono también.
Well, I can start speaking loud but it's going to go up. So let me know if you I can wait a little bit until shall I not wait? Anyway, can you hear me now?
Yes.
Yeah, okay. So thank you for the invitation, Bruce, and all of you guys, thank you for being here. The title of my talk is Journey to Africa, and I was kind of going to start with explaining what is why I gave this title. So, which means I'm going to talk a little bit about myself, but also talk about the project that we're working together with Petra and as well. So I did environmental engineering. And as an American student, I came to England, and I quite liked research. And they were asking me whether I wanted to do a PhD. And after a few months, I kind of gave in and I said, well, let's try. And I applied for a project which I didn't know anything about, but I really liked the topic. And it ended up being a PhD at the University of Leeds here in England. And it was a European project. So how much, guys, do you know about European projects and European funding? Yeah. Have you ever worked in any? Okay, so you know, some of it might be something that you already know, and some of it will be perhaps new. So I will talk about European projects because I did my PhD in a European project and I really liked it. It was a really interesting journey. It was a really interesting experience. I met with a lot of people. I did a lot of networking. The science was really interesting too as well, of course. And because there was a great amount of funding to be able to do big science, I did a lot of synchronous work. I don't know if that is anything that you know about, but it was just really high-end research in a way. So it gave a lot of opportunities, but I also, as I said, I was involved with a lot of people, working with them and networking with them, and within the network, moving in between the universities. So we had 5 partner universities, and because of that, I did part of my PhD in Denmark as well. So it was just a really good experience, and it happened that I didn't continue in a European project at that time, but part of the title of the topic is that it's something that I like to advocate, something that I like to positively talk about and support, because I think European funding and European projects are really good. And then I did work in other projects as well, so I have experience with research council projects, but still European is something that I feel close to, interesting to my heart. So I work as a research associate in Sheffield, but about a year and a half ago, my boss came to me as whether I wanted to manage this European project, which was this advocate project. So that's the other part of the title, that is the advocate project we're going to talk about, and my journey through it in a way, or just the project's journey in a way. So I work in research, but I also work in project management and helping these guys along the background, I guess. And I also do mentoring and coaching at the university. So I find it important to help people's mindsets through research, how they, it's not just what they do, but who they are, I guess, along the journey, and who you want to be. So what I wanted to talk to you about today is the advocate project itself. Its scientific and commercial value, and what European research and what European funding brings into our society and to our economics, not just in the EU, but worldwide. And also because EU funding, and well, actually, I didn't mention that it's an initial training network. So again, is that familiar? So it's supposed to give a lot of training to the fellows that helps them along, not just in science, but in their career as well. So it's also investing in personal and career development. And so just through the advocate project, we kind of go through these points and see how they come out and how they work in EU funding. So a little bit more about the advocate project. Ruth mentioned already. The project is working on developing sustainable in situ remediation methods for groundwater and soil contamination. So that was remediation. Are you familiar with the words remediation? So remediation, okay. Some says yes, some says no, some is going to explain it. So remediation is basically bringing it back into its original state, original status. So if there's a contaminated water, medium water system from industrial activities, for example, then how we can bring it back. Or if you just think about wastewater, for example, that's something that we constantly remediate, we bring it back to it, and then we bring it back into the circulation. And there was also in situ. Is that familiar? Yeah. And the other thing I wanted to groundwater. Is that okay? Just so you know where we are. So the coordinator of the project is the University of Sheffield, and the principal investigator is in the University of Sheffield as well. We have 8 other partners who are beneficiaries, so basically they receive funding through their fellows and through their research activities. So they across Europe, which means Poland, Germany, Netherlands, Belgium, sorry, and Switzerland. There are 14 fellows, and they are kind of, they've been from all over the world, really, from South America to New Zealand. And there's also 12 industrial partners, industrial associates, which means that the students have, or the fellows have the opportunity to interact with them and get feedback from them. But also these companies are interested to get the results of the research firsthand and hopefully integrate it into their work. So it's a Marie Curie Actions funded European funding and initial training network, as I mentioned. And it's been running from April 2011, so it's ending very soon, in the next few months, basically. So we had 4 years of research, and I came into the project in 2013, so I came into it fairly late. And yeah, approximately €3.5 million, which is quite a nice amount to to spend in 4 years, but with 8 partners, it's about 9 partners altogether. It's hopefully well spent. So the Advocate project was written according to the Water Framework Directive in the EU at that time. So there's actually an expectation from the EU to provide resources that they can integrate into their policies and into their further work. So a little bit of the background. Basically, it's just so important, not just in the EU, everywhere, that groundwater and surface water and soil basically gives us food and water. So, and the more the population is growing, the more the need there is to have clean water and food. And in the EU, at that time, that was 4 years ago, there was an estimated 39 billion euros that would be needed to contaminate all the sites that has, let me try and get it right. So they have contaminating potential. So they are industrial sites where if anything happens, some of them has already causing contamination in groundwater, soil and surface water, fresh water. But others have the potential and poses risk towards these ecosystems. So so the sources are industrial activities, factories, production, increased social and economical demand. There can be leakages, accidents, or technologies with treatment waste production. There's all sorts of problems and all sorts of strains on this demand. And so, maybe think about it in a way that you have a big stack of paper, and every day you deal with 5 paper, and you put on 7 more. So it's not just that there was that 39 billion euros that we would need at that time to clean those sites, but every day it's being added to because these activities keep going. So where research comes in and where funding comes in and European funding comes in as well, is that we do what industry doesn't necessarily have time for or the resources for to do. I mean, they could have, but academia, a lot of these resources are already there, a lot of the knowledge and the science is already there. So it really works working together, and that's more and more the future. So I don't have that many slides actually, but I have 2 or 3 slides now on outcomes. And these outcomes, this slide is about what are the outcomes for the EU, for our partners, partner institutions and industrial partners. And basically with the technologies, with the methodologies, our partners, and we've been working on, what we try to do is have more reliable, accurate and relatively fast quantification of the extent of contaminations. So these 39 billion euros, how did we make this number? How did it come to life? Whether it's accurate or how much is accurate, so trying to make it more accurate. Measure up the sites really and their contaminating potential. So it's really contaminant and site focused because every site and every contamination, every scenario and every situation is going to be different. So which can mean a lot more money, but if you do it cleverly, we can be more focused with using not more money, or hopefully being more cost effective and sustainable in our remediation processes. So we've been working on computer models and field processes as well. We also try to establish alternative technologies, part of which is natural attenuation, which is basically using nature's power to do some of the work for us. So Petra, we were talking about that, how to use biology that's already there in the environment to help remediate contaminated sites. And then, of course, a big part of the project as well and management is that, and there's a whole work package on it, which is to take this message to industry, to the government and to the public to try and reshape perhaps the opinion where it needs to be. It doesn't have to be everywhere. And then to try and help inform them educated decision making and introduce change where it needs to be. I put down outcomes from the fellows' point of view. I mean, you can really talk to them to ask them what they think. But this is how I see it from my perspective and from my experience as a fellow when I was one. And I know it's a busy way of doing your PhDs and doing your projects because there's a lot of feedback you need to give to the project and to the EU. There's a lot of paperwork involved and a lot of pressure on the way, hopefully not always negative. But it also comes with a lot of scientific and professional training. So when I went through my PhD, there was a lot of other PhD students who were on in basically different funding, different projects, and they had much less of the opportunities that I had. So I could go to conferences and I could take a message of what I was doing around and really talk to people. And I also had a great support from the network because we had so many meetings, so we had, and it was similar in the Ethical project as well, at least 2 meetings per year, where we got together for a few days for a whole week, and we shared the work we were doing. We learned something, so there was a demand. I definitely wanted to learn about geochemical processes, and that was the topic, and then the scientists came, senior scientists came and taught us. We went for field works and then we went for drinks in the evening, so it was like a 24/7 interaction, but it was a great support, it was a great environment altogether. So I think, yeah, the difference I really found in advocates, and as the EU funding is changing, that there's a much more demand for industrial interaction. So we're not doing research on the academic level, but we're actually doing research on the global, on the industrial level as well. I mean, it might be different for you with biomedical sciences. I think some of you are from there. But in terms of environmental science and environmental engineering, it's still something that's growing. And in the Advocate project, there was much more opportunity than I noticed. We didn't have any industrial partners, so I never had the opportunity to go and work with them. But it was different now, and it's becoming more and more different and more available for people. So then they can decide, one, they can transfer the knowledge to industry, back and forth really, but then they can decide whether they want to work in research or research and development or whatever they want to do. So it's a career development for them as well. So that was I was thinking to find something that kind of reflects how I personally feel about the whole process. And I quite like this picture. It's from, I think it's from Personal Development website. I quite like the power of nature. That was one of the things that I mentioned, that we're trying to use the power of nature to help our decontamination processes. And also, it reflects the beauty of nature, which means that we should be in synchron with it. So perhaps we don't always have to put 7 more papers on that stack, but we can just put 1 and maybe go with more as well. And I quite like the image of the 2 guys, one helping the other one, because I think it's nice for everyone to be up there, including fellows, and enjoy the view together. And so, in a way, it's about how we impact and how we empower the students who grew out from this project as well. So that when they make the decisions and when they get to the point of making these decisions, they will have that inspiration behind them. That's it. Any questions? I wasn't scientific that much, actually.
Okay, so basically, your role is some companies come to you or some government comes to you, tells you there is some groundwater here. We want you to analyze it and check how much contaminated it is and how we can make the most use of it. I mean, there's been a couple of slides that I may have missed, but I'm trying to understand what advocate exactly is. I mean, what your actual research is about.
So the actual, yeah, sorry. I mean, that's where advocate came from. So Advancing Sustainable Institute.
So it's more the way how it works. I mean, it is coordinated by the University of Sheffield, but you go to the ground field and see, check where groundwater is,
Yes.
and you know, study the field to, I don't know, design how to best make use of that groundwater.
I think it's more step back from that. Like there already are consultants that are there, like in the field, working in this industry to clean up the field. So the job of the research is more to improve the technologies that these consultants would have when they go talk to the industry and say, okay, this is what we can do, this is how we'll measure it. So it's just to improve the tools for the on the ground.
So basically for the project, when you submit it, it's like a 40-page document, and you outline most of. So you already have your partners, and you already have the contaminated sites that you're going to be looking at. So whether someone comes to you or you go to them and say, well, this is what we'd like to do, would you like to work together, would you like to give us that site to work on as well.
Okay, so the sites have been previously identified, right?
Yeah, so it's not like we go and find the sites,
Okay.
but at the same time, based on the sites that we have and based on the contaminant processes we look at, one of the work packages develops like a model that then you can use to get this information, like how much it would, if there's a contaminated site, how much it would take for it to decontaminate, what would be the best process, how risky it is that the contamination would occur. If it occurs, what's the medium around it, so what the soil layer, where the groundwater is, how deep, so to try and predict those things better as well, based on using sites that are already contaminated.
I'm talking. I know nothing about this. Okay, so what's the main client, the main customer?
I mean, yeah, that's why I kind of put it that it's science, industry, socio-economic, the EU. I mean, the main customer is the EU in the end, because they are the ones who give you 3.5 million euros, which is a huge amount of money. So you're not just giving back peanuts to them or anything like that. So yeah, so I imagine they are the main customers actually, who they'll be using. So we have in some of our deliverables are policy guidance documents. So that goes back towards the EU. It feeds back into their policy making and decision making of, you know, what are the main projects, what are the important things to look at in the EU. What is it that we should be funding? What is it that we should be giving more money to?
But what is the situation? Because I guess like the industry is the one doing the contamination. And now the EU and this water frame is imposing a minimum of clean, they have to be clean or they have to clean back, so they are the one getting the benefit. I mean, if not, they will be fine. Or what's the situation at the moment? The industry contaminates the water and they are responsible, I guess. So at the end, they are the kind for this week, the other one being benefited.
So you mean if they end up in a situation just
No, I mean that they are the one contaminating industries.
Yes, so
And they are being imposed by US a minimum of contamination, so they are the one getting the benefit of the reason being done. No?
you say like the way the situation would be in most countries in Europe is that there's some guidelines for how clean water has to be, right? So if you have contaminated water under your property, you're responsible for cleaning it to a certain level. But then the other people involved, so there would be the company or the people that own this land, or one of the people that would want to have technology that will actually make it clean. Then there usually would be the government regulators that are figuring out ways to measure what clean actually means. Like, it's not so easy maybe, like which contaminations is important, what levels you want, whatever. So the government would be involved at that level. And then you also have maybe a private company that would be contracted to actually do the thing out. So in the end, I think
But they
there's kind
still
of,
didn't want pain, or no? Because
Yeah,
they
they
want the
but
cost.
if they go bankrupt, then also the government would be paying. So it depends.
So the
But in
point
the
is
end, I
not
think there's three industries really involved. There's the government involved, and there's kind of like consultants involved, and there's
so the balance between them, because then with the technologies and the models developed, it goes back to the industrial partners, so they can improve on their technologies, so they can be learn.
it's like plantation and a doctor and a pharmaceutical company. We're like the pharmaceutical company.
Yeah, no, I'm just considering you do the contamination, you don't get fined otherwise, no? If you don't.
I mean, I just want to say something like with the Aquabate project, what we're looking is to find technologies that would help us to clean the mess, because at the end of the day we have a lot of contamination in soil and in groundwater. So this is this initiative from the European Union, and what we do, we are a multidisciplinary team that comes from 4 countries, and we're trying to, I mean, we have different backgrounds, so all together we're trying to find a solution for this. And in some cases is to find the remediation, how to find the remediation, and in other cases is to find how to find the solution. It's really like this. So at the end of the day, it's industries, the European Union, and us, because the contamination is there, and we will.
But I think like the state of the world. But like right now, there's the amount of money that goes into industry for making new things compared to the amount of money that goes into figuring out how to clean those things up or not make pollution when you're making those things. I mean, the imbalance is astronomical. So the state of things now is that we don't actually know, or industry does not know or have the capacity not to pollute. So that would be part of our research, would be to be able to at least say, like, to try to make ways to avoid, because there really isn't right now, or to make ways to clean it up, because there really isn't. And I would also say that the fines that these industries pay it's a big company, it's like nothing. And I mean, the cost, because they don't know how to clean it up, it's like, okay, you pay €1,000 every 6 months, or you invest €1 million in trying to figure out some new scientific problem. So,
but also you don't have to be, so in this case when research and academia is involved, that's why I was mentioning that the company doesn't have to pay for research and development themselves, but the university does it for them. So they can get the benefits of it. And then maybe they can get to that level that they don't choose to pay the fine, but they say, well, we have a good enough technology, and in the name of sustainability and keeping things at a better level, they might choose in the future.
And if you do develop a particular technology, it is the University of Sheffield the one filling up the patent, or it is the partner company? Or?
Oh, that's a very good question actually, and I think I should know quite a bit of that. But in this case, the patents belong to the university. I think it's, at least that's how it is in the UK, and I imagine it's going to be different EU-wise.
Okay. But it would be a kind of revenue for the university to be, besides doing the proper research and getting the funding from the EU, if they develop this patent, the company later on will need to use that patent.
Well, see, that I mean, there's a contract. So your question was whether the company would use it, or but that patent would belong to the University of Sheffield. So in that case, because the companies are partners on an information level and on a knowledge transfer level, but they are not beneficiaries in these projects. So in the end, they don't financially they're not involved. Unless, I mean, if you go there as a trainee, then
no. But in the end, it is a very good project to be done by the University of Sheffield, because the companies don't invest in research and development. they partner with you guys, so you develop the technology, you fill up the patent, and then they start using that patent, and they will pay you for the use of it. So it's
the University of Sheffield. I mean, the companies normally have these things in place as well, but the University of Sheffield will do it very well.
Yes, I have one question. This project is about to finish, so normally these IT networks, you reapply again for more funding. Is that the case of this advocate project, or will you stop here?
We're not going to continue with exactly the same project,
Okay.
but there are outcomes from the project.
Okay.
In terms of how things should be implemented or how things could be changed. We've applied for another one, which is not going to be the continuation, because normally, I mean, rarely, it really depends how it goes and what the situation is, but you rarely actually continue the same, because you have to submit applications before. I mean, when I was doing mine, we had another network coming on just a year after, so we were kind of working parallel, similar universities, same universities in water. It really depends, and it really depends what the outcomes are.
Yeah, I'm talking about the outcomes. In these 15 PhDs, which will be for you the more the candidates to go to the market? So which is the main outcome, the one you will highlight?
Sorry, I don't understand.
In these 15 PhD, there's always one that has very good results, and these results can be applied in the market. So what will be? Is this a model? Is this a remediation technique? Is this, which is for you the project that is the best one?
That's a challenging question, actually, because I wouldn't want to name one person within, and I think that.
Well, you don't need to name the person, but the project.
I think there's actually a few really nice technologies and really nice ways of doing things that's coming out. I mean, the project Natalia is going to talk about is very, very interesting, and I think you'll really find more of the answer in that as well. And then some of the modeling has been quite good. So both on the field side and both on the software side and modeling side, I think there are good outcomes. Yeah.
Okay, thank you.
Hi everybody. So I'm Petra. I'm on the Advocate project, so you'll know more about one of the actual project which was solved within the Advocate. And I'm going to talk about bacteria, as Ruth said. So it's about bacterial batteries. And I'm looking into ways how to produce electricity from pollution, and also how to enhance the bioremediation we were talking about. So there we go. Okay, so it's about electricity. I'm sure you know what it is, but I've got a definition in there, just if you weren't sure at the time. But I'm sure you know lightnings. So when there's different charge between the clouds and the Earth, we can see actually electricity ourselves in the form of lightning. Or for me personally, the easiest way to imagine electricity is to imagine a wire and tiny particles called electrons moving through it. So we're going to talk about electrons a lot, because remediation and bioremediation specifically with bacteria is about electrons. So it's about bacteria. We're going to talk about bacteria metabolism, how they work, what they eat and how they process it, and where the electrons come from this. in this case. So, there's a baseball player, it's not a bacterium. Bacteria are very similar to people in certain ways. So, when the baseball player wants to play his game, he eats something, he eats food, I do sugar in there. And when you eat the sugar, you degrade it into carbon dioxide, which I'm sure you know that you breathe it out when you breathe. And also within your body, you'll find hydrogen protons and electrons. So there they are, electrons. And you breathe in oxygen. And that oxygen reacts with the protons and the electrons, and it forms water within your body. So that's what we do all the time. And this is how a bacterium does it. It's actually the same process. So a bacterium eats sugar, degrades it into carbon dioxide, protons, electrons, and it can use oxygen and form water. What's really clever about bacteria is that they don't have to eat nice things like sugars and proteins and fats we eat, but they can eat some nasty stuff. So on the picture there's benzene, but there's lots of other contamination. The bacteria are really versatile because we've been contaminating this earth like, let's say, last 100 years. But they are really fast in developing what they can eat because that gives them the advantage in the environment because they can get more food. So they're like, oh, let's eat something else and just nice things. So they develop the ability to eat their stuff. Because you know they multiply quite quickly. You can have half an hour and then in another half an hour you're going to have 2 bacteria out of 2. So this process is really quick, whereas people take like 20 years to multiply. So, yeah. So they know how to eat their stuff. How's that useful? Well, we're going to talk about bacterial batteries. A posh name for this is a microbial fuel cell, if you ever wanted to read any literature about it. And this is how they work. It's practically separating the 2 reactions. So here we go. Here's our sugar, which goes to carbon dioxide in a battery. So here we go. We've got 2 electrodes there. One of them doesn't have access to oxygen. That's the one on the left side, where the bacteria are. The other one has access to oxygen. And these 2 are separated by a semi-parallel separator. So it can transport certain things, but can't transport the others. And then this is our battery. So it's got electrodes in there, and when you put bacteria in it, they attach to the electrode. And as I said, in that chamber where they don't have any oxygen. So they eat that sugar, they make carbon dioxide from it, and they make protons and hydrogen protons and electrons. And then what happens, because it's in the battery, the electrons go to the electrode and then are transported through the wires. And as I said, the membrane, the separator can transport certain things. So in this case, it's the hydrogen proton which goes through the membrane. And here we go, the light bulb is shining. So there's a bit of electricity coming out from that process. And the other chamber has got access to oxygen, which then again reacts with electrons and protons and forms water. So you can see that this process is now not happening in the bacterium only itself, but it's separated into 2 chambers, and that's quite useful because we can then get the electricity out of it. And again, it doesn't have to be just sugar what bacteria eat, but it can be our contamination we were talking about. My project specifically is focused on groundwater pollution. Gabby was already talking about groundwater. I've got a picture about it. That's the water beneath our feet. It flows between the fractures and pores of the rocks, as it's there on the picture. And it's an important source of drinking water. I work in Sheffield. In there we get water from the dams, but in here in London, the water comes from the aquifer beneath London. So you're drinking groundwater in here. Yeah, England, Wales, 30% of drinking water comes from groundwater. It varies depending on where you are in the UK. So the water flows beneath the ground in a certain direction, and then we can have a well and then pump it out and get our drinking water. But there can be a problem with it. So we're talking about industry. I've got a factory there with smoke and nice contamination plume, but it can be such a simple thing like a petrol station. So under the petrol station, you know that there's the petrol tanks which store the petrol. People install it there. This stuff can get rusty. It can leak. Sometimes people don't even dig out the old tank and then just put their new one. So all this fuel can get into water. I'm sure you don't want to be drinking that. So, yeah, there's this lecturer, Professor David Lerner at the University of Sheffield, and they say that wherever he goes, there's contamination. But what he says that practically every petrol station has got a contamination problem. So, there you go. That's our groundwater. And there's our contamination plume. And as I said, bacteria have got the ability to eat this pollution, which is great. But sometimes they can't do it fast enough. So you saw in the picture that when you eat something, you need the oxygen to pass the electrons on. So what people sometimes do, you drill a well and you pump air under the ground, so you give bacteria oxygen to breathe it, so they can actually eat this, because if they eat it and they can't pass the electrons up to something else, that they're not going to really do it. So that's one way to do it. But pumping air under the ground, it uses lots of electricity, and it's a bit difficult to transport the oxygen beneath the ground into all the places. So I'm looking into finding some other solution how to provide bacteria with their breathing. So here we go. That's our bacteria battery. So I'm developing a technology where we could install an electrode under the ground. The bacteria could attach to it. They could eat the contamination. I'll look specifically into the phenols, there's a structure on the screen. So they could degrade it into carbon dioxide, and as they do it, they pass electrons onto the electrode. It produces a bit of electricity, and it reacts with oxygen somewhere else. So it kind of eliminates this whole transfer of oxygen underground, and you can have the bacteria interacting with the electrode under the ground. And the amount of electricity produced from this is really small. You wouldn't power a house from it. I often get a question like this, so no, you wouldn't. But the advantage is, as you remember, I previously said, we need to use often electricity to do the remediation. So if we don't have to, that's the advantage. So it's more like a passive system where you don't need to provide electricity to do stuff. So this is how it looks like in the lab. I've got a tiny system because it's just this is just the first experiment, because this field is really new. People started doing research into the bacterial batteries like 10 years ago, so it's relatively new. And this field for bioremediation is really, really new. That's even That's even more. So I've got this little system where I've got the contaminated water and I've got the electrodes in there, and I've got 0.1 volts. Yay! But it kind of works. So yeah, there you go. If you've got any questions.
Do you know anyone in Spain doing more or less the same research?
No, I don't know about. I know about Italians. I know about Italians that are doing. They're doing different contamination, but they do the same topic. And then regarding generally the bacterial batteries, there's a big group in Netherlands, I think. Yeah.
Do you make any use of the dioxide of carbon dioxide?
Carbon dioxide? No, no, that's just a byproduct of the metabolism.
This bacteria can live in high rates of temperatures, or just a limited range?
Oh, well, in this case, because it's groundwater, that's usually around 12 degrees Celsius. And the bacteria, when they live somewhere, they adapt to those conditions. So the ones which are in the groundwater, they like the 12 degrees. But often when you do something in the lab, it's 25-ish. And the bacteria are a bit faster. So all the reactions get a bit faster. But hope that answers the question.
Okay, yeah, sure.
Yeah.
What bacteria are they? And do you add them to the water, or are the bacteria that are already?
That's a good question. I didn't say that. Yeah. So in general, in water, there's always bacteria because they're everywhere. They're just, yeah, they're just amazing. Like there's even like in Yosemite, I think in Yellowstone in the US, they live in acids. They can do anything. But when you've got the contamination, there's more bacteria in it than in the clean water because they've got more fluid in there. So we are actually using the bacteria which are already there. And it's different species, it's like a big community, so they work together, they kind of cooperate. So one of them eats something and then produces something, and then the other one eats it as a food. And so, yeah.
But isn't the community usually much simpler, the bacterial community, when you have a lot of contamination? It's a pretty big stressor. So does that, you don't have any sequencing data? Or do you?
I do. Well, what I did, I took it from the fringe of the plume.
Okay.
So it's not the hardcore contaminated stuff. It's from the fringe. So,
Why do the bacteria attach to the electrode?
bacteria like attaching to things. It does give them the advantage to survive. So when they're limited with their food or anything, they can live like a little community, and if the top layer dies, then just the bottom survives. So they will be on the sediment under the ground, so it will be like a natural process. And then there's specific species which actually like producing electricity, so they are very likely to attach to the electrode because it's easy for them to use it.
So what do you think is the main limitation of this sort of batteries?
The limitation can be there's a distance between the two electrodes, for example. So I think the biggest limitation is now kind of to engineer the system in the way that it can work. So you might have to put the electrodes quite close together so the resistance will be small. And I think another one is that we don't know much about how these natural bacteria will interact with the system. So I've read papers and they usually use like single bacterium, but you don't have that in the field. You've got like a community, and then they use single contaminant, single chemical, but I've got a mixture. So yeah, it's just lack of kind of field type of data.
So based on what you've learned in the few years. Do you think it works investing into engineering field scale application?
I think there's still much more research needed on the lab scale first. And in my case, I think it would be worth trying to simulate in the lab the field condition. So build like a little aquifer with the sand in it and then put the water in it and try to see how that works first. I'm optimistic, I think it can work. But
So in the large scale, what, like physically, how, like, what it would be?
so I'm
Because you have to be real into it, so being really like a tube.
Yeah. So on the field scale, I think you would try to sort of build like a medium. The electrode would be like something which the water can flow through. So as it flows through the grains, you would try to build the electrode in the like grains. So it would be like probably carbon, and then it would flow through it. And so that's how we would do it, and it would be quite big. So for example, for this field I'm working on, the plume is, if I go back to this, so the width of the plume is about 25 meters. Yeah. But then again, because it's not research, it might be that it might only work only on small plumes.
But you get like an area there. Like,
Yeah, it's so. I'm not sure about the width of the plume. It's going to be at least 10 meters.
well,
But it's 25 meters.
how are you gonna get that inside?
You dig a trench,
Like a huge.
yeah, and then you shove the material in. And connect it to the wires.
These kind of barriers are already being built in use. I mean, not
Huge.
for electricity production, but filters that can filter the water.
So it's not very deep groundwater.
It is.
Wow, very expensive.
Yeah.
And what type of industry is the one that contaminates the most? I mean, yeah, but
Chemical industry.
Yeah.
And petrochemical industry is.
and these industries have like this plant where they put all the contaminated products,
It's
like
usually-
garbage.
It's sometimes they have like a lake where they dump stuff,
Yeah.
or sometimes they have an accident. That's it. It leaks.
Yeah,
and that's it. You know. So it can be, sometimes like they ignore the health and safety stuff and they just like chuck stuff on the ground and that's it.
Yeah.
It's going to go into groundwater. Well, it's not good.
No, very good.
But I was just going to tell you that in Germany, at least, the largest owner of contaminated land is Deutsche Bahn. So the national train system owns more contaminated land than anyone else. So yeah, I mean, there's so
In the US, it's the US military, but
they
it's
do a lot of testing.
but
There's a lot of like testing of weapons. Makes sense.
yeah, but it's usually like the fuel. It's the fuel for the planes, or it's for the trains. It's getting fuel related.
Have you ever seen it? I think it's on YouTube. The nuclear developments through the world. So it has like a map of the world, and it goes through the 20th century, so 1900s, and it shows like in each year which country where they set off the bomb. Have you seen that one? Yeah, and then it goes in, it's crazy. So there you go. That's just testing.
This must be compared to all the other testing. It's supposed to be like I don't know. But one other problem with doing this kind of research is that often you don't have access to all of the information of what the contaminant is. Because if it's a private company, they're very proprietary and won't actually tell you what pollution is there and why it's there.
Okay.
The last one, so I'm going to introduce myself. My name is Natalia, and I'm a PhD student from the University of Liège in Belgium. And thank you, Ruth, for inviting me. And I'm so happy to be here because I think that the BUP is really the place where the craziest and the best ideas for scientists come up. So I'm so happy to be here tonight. I'm going to talk tonight about how we can improve, how can we develop technologies or what we can see below us, because this is really, there's something going on and we don't know how to solve it. So I wanted to start with this picture because I really love it, because when people ask me what you do as a scientist and I show them this, they get really confused. They ask me if I do Chinese New Year celebrations or something like this, or what's that. So I think that hopefully in the next 15 minutes you will understand what is this about. And this is how I'm going to do it. I'm going to try to work with it. Okay. I'm going to talk about the problem with this. We have some problems with the underground, and I would like to talk about why and who cares. And then I'm going to talk about this technology that we have developed, and we install it in our contaminated sites. And I want to show you as well about some information, the kind of information that we get, and get some conclusions. So hopefully you will be happy. I'm going to start with the scenery. Okay, so we have an industrial site, we have some industrial activities, and as a consequence of that, we can get some contamination. And in this case, we have contaminated waste, which is this pile of stuff which is laying over soil and the ground. So if we take a knife and we make a cut and we see the profile, this is what we would see in this case. In the very bottom, we have the groundwater, and then we have an area between the soil and the groundwater. And this area, it's called the barrier zone. And it's really important because just imagine that it's raining and the contaminants are leaching and they're going towards the groundwater. The barrier zone is going to be the place that will control all this release of contamination. And you might think, okay, so what's the point of all this? Okay, let's imagine that I moved there because I love it. And let's imagine that I'm there and I'm going to have a double exposure, because the contaminants are traveling there. Well, first of all, there could be some chemical reactions and there could be some volatiles, so that would go up and I will be exposed. And also, well, as Petra said, many countries, most of the drinking water, they come from the groundwater. So, we really need to find a solution. And I will not be the only one who will be in trouble. You will be in trouble and the ecosystem will be in trouble. Okay, so we need to really understand how this contamination is going down if we want to, to clean the mess and if we want to protect what we have. So this is where my PhD project comes along, and what we want to do is we want to develop a model where we can understand how this contamination is distributing in this area, and also how this is split, mixing with the groundwater. But because in Belgium we're really brave, we want to do it directly with field conditions. We want to create something that we can put directly in the field and understand how it works. And it sounds fantastic, but I'm going to talk about the starting point, and I want to use a comparison so you can understand how the challenge was for me. So, okay, I saw this and I thought, okay, great, I'm going to say the word, but the thing is, for example, most of you would know more than I do. Like when we get sick, there is always a way to see what's inside of us, because there are some technologies that allows us to see through. And eventually, if you're really lucky, then there is a solution for the problem. But what happens when you're in the field and you see all this contaminated soil? I took that photo. I remember that day I was looking down and I was thinking, oh my gosh, I can't see anything. This is going to be a long PhD because I have no idea how I'm going to start this, okay? So I decided to try to find a solution, to look for technologies that could measure, for example, the flux of contamination going down directly in the field. And after several months, I realized that I found out that there was a limited application of these technologies. And why is that? Because most of the technologies, they have been developed for agricultural sites. Well, I don't know if you're drunk or not, but this and this, they have nothing to do with each other, okay? Because, well, in an agricultural field, you can have a certain type of soil, but at industrial sites, you can have artificial soil. And also, you have different types of contamination. Like, at industrial sites, you have mainly pesticides. At agricultural sites, sorry. And at industrial sites, you have God knows what. Because I've seen it, you know. And also, the depth of investigation is completely different. Because for the people who work in agriculture, they only care about the first meters of soil. But when we have this big problem, it could go meters and meters down. So basically, to make a comparison, it's like we want to see the bone, but we can only reach the skin with our technologies, right? But that was really discouraging, but I didn't give up, and I kept looking for something, and finally, we found some sort of an alternative. And this is what we call the Bacterial experimental setup. So I'm going to explain it. It has two parts, okay? So the first part, it's called the Bacterial monitoring system. So what is this? This is something that some scientists in Israel have developed. They have developed this, and it's basically an inclined borehole which contains sensors along its depth. So with these sensors, we will understand the water that is going down, and also we will be able to see the chemistry. And why are we using an inclined borehole instead of a normal vertical borehole? Well, we want to study what is coming from above. So if we make a vertical borehole, what is coming from above is going to be disturbed by the effects of drilling. So I saw this and I thought this is just brilliant. So for our project, we would like to have 2 inclined boreholes, and we would like to install 3 types of sensors. Okay? So the first sensor is going to be distributed at different depths, and it's going to measure the water at different depths. Okay? Then the second one is called biosampling ports, and this would allow us to get some chemical information from the matrix, from the material of the soil. And because we knew that in our site we would have some fractures, we decided to install another sensor, another type of sensor, where we could get the water that was going through the fractures. So this is the first part. It's basically 2 inclined boreholes with sensors. Then the second part is called cross-hole geophysics. So with this, what we want to have, we want to characterize, we want to see what's inside. And how do we do this? Okay, the concept is relatively easy. So we have the ground, and then we drill 2 vertical boreholes. And we install electrodes in the boreholes. So what we do is we inject an electrical current, we measure the voltage, and we get an image of the electrical properties of the surface. So it's like an image, so we can understand, we can see the structure, and we can understand what's going on. So we can do this underground characterization. Okay, so we have inclined boreholes and we have vertical boreholes. And what we wanted to do was the following. We wanted to drill the inclined boreholes, then we wanted to drill the vertical boreholes, then we wanted to leave the site as it is and wait for the rain. And this is what we call natural recharge conditions. We wanted to see if the sensor would work, if everything was working. And eventually, if everything was working, we wanted to do an experiment. So what we wanted to do is to pour in the ground a tracer, which is a substance that you mix with the fluids, and you use this technology to trap it down. So what was the kind of results that we wanted to get? We wanted to get like a 3D image of the subsurface, so we can understand it, and we wanted to use the data from the inclined boreholes, so at the end we could characterize what's below us, right? Okay, so now I'm going to talk about the site. The site, it's used to be a coke plant, and it's located in the western part of Belgium. And just with this picture, with the Google map image, you can already see the contamination. Do you see this blue part? This is cyanide in the soil. It's not the only contaminant. We have heavy metals in the soil as well, and also we have benzene. I mean, the place is the closest to hell I've been to, seriously. And it's really contaminated. And in this place we have artificial ground, we have sand, we have chalk, and then this battle zone is 8 meter. It has a thickness of 8 meter. So now I'm going to show you some pictures about how to install the stuff. And I will start with the inclined boreholes, okay? So believe it or not, all the sensors that we were talking about, they are installed in these two sleeves. This is what we call the flexible sleeves, okay? So we went to the fields and we took this sleeve with us. And while we were drilling these inclined boreholes, we were preparing the sleeves, that's me. And then once the sleeves were ready, we took them and then we installed them in the inclined borehole. So this is for the inclined borehole part. And then for the vertical boreholes, we had to build everything at the university, and I have to say it. So we were building everything in the university, and we had to have the tubes, and we were building the adapters, and this was really a team effort. And we had everything built, and then we went to the fields, and we drilled the vertical boreholes. And once the vertical boreholes were drilled, we just put the stuff inside. Okay. So this is what we have right now, what we have installed in the field. So we have these two inclined boreholes with sensors, and then we have the vertical boreholes where we can get the images of the subsurface. Okay. So this is the thing. We Okay, so we have all these installed in the field. We said, okay, we're going to dig it like this. We're going to wait for the rain to come, and we're going to see if this is working or not, because we might have installed something and nothing happens. So I'm going to show you an example from one borehole. We were collecting data from 3 places. First of all, we were collecting the rain. And what you can see here is the amount of rain that was falling down over this time. And then we also have some wells, and we had some sensors, so we could see how far was the groundwater from the ground. So now, if the system was working, we had 6 sensors that would measure the water content, right? So if the system would work, we should have 6 lines that show us the water content at different depths. And what is more, if the rain was infiltrating in the ground, these lines wouldn't be flat, they would move. But we don't know if that worked or not. Did it work? Yes, it did. So we have 6 lines that reflect the water that is coming, and the 6 lines are not flat. We have, for example, these 2 lines correspond to the sensors which are in shallow depths, and as you can see, they really move fast because the water is coming down and it's infiltrating. And once the water is infiltrating, then water is starting to move in the fractures of the ground. So it's pretty good because we already see what's going on. And also, we were capable of taking some samples. So we were analyzing the chemistry of the samples, and we noticed when we were using this diagram to understand the chemistry, to see what is the chemistry of each sample. And we noticed that the shallow samples, they were different chemically from the deeper ones. And furthermore, we observed that when the rain was infiltrating, the chemistry was further changing. So this is really important to take into account when you have a contaminant, because there could be an evolution. So this is from the inclined borehole part. Okay, but we also had the vertical boreholes, and this is what we did. We had 4 boreholes, so we were taking images in perspective of boreholes, and if we have 4 boreholes, we could take 6 images, and finally, we could build more or less an image of what's underground. So far so good. We thought, okay, this is working, so now we can do an experiment. So we decided to do an infiltration experiment. So the objective was to build a pond and to pour a tracer, and then to use these vertical boreholes to get an image and see what's happening. So this is what we did. This is the pond and the tracer, and this is diagram that shows more or less what it looks like. It's the tracer falling. Now this is one picture, but because this was working really well, we thought, okay, why don't we, as we're putting the tracer, why don't we put 2 lines of electrodes in the surface, and we inject a current, and we do the same with the boreholes, but in the surface, because this could give us a better image, a better resolution of what's happening near the surface. So this is what we did. This is the infiltration point where we had the tracer, so we built a line of electrodes here, and we connected cables, and we put the electrical current. And what you can see here is the evolution of the tracer toward, with the time. And we did the same in the other direction. And well, this is what it looks like. So this is basically the end of my talk. My conclusion is that we came up with this idea where we could, with this system, we can understand, we can see the water that is falling down in this fallow zone. We can also get a bit of a hint of the chemistry, and also we can get images of the substance. And that's it. Thank you very much.
Just one question. What you obtain is a 2D image, right?
Yes,
so between
yes,
two electrodes.
And we are assembling it. I mean, this is a 2D version of the images, and then well, we're thinking about doing a 3D image. But the idea was to see more or less in 3D what's going on.
And the distance is 4 meters, right? Between the.
It depends. The distance between the boreholes.
Yes.
Yeah, it depends, because we were going to do something really straight, but we have to modify it. So it's like 3 meters or 5 meters. So we have, yes.
Okay. And just finally, one question: how do you obtain the chemistry? So how do you obtain which chemical compounds you find in them?
You mean the way that we retrieve the sample?
Yeah, it's just resistivity. You mentioned resistivity, but is this the way how you correlate your signal with the chemical compound?
Well, for the geophysics.
Because you are measuring the tracer, so this is the only thing you have.
Yes, but the way we measure the tracer is not with chemicals, it's with the difference in resistivity. So we choose something, the tracer that we chose is, it has an electrical conductivity which was like really, really different from the ground. So we're using difference in resistivity in order to get that, but I didn't explain it here because I didn't know if you would be familiar with that.
I think everybody is hungry.
Yeah. Thank you.
Día a día.